Hello everyone, myself Mohd Saad, and today we are going to take a very interesting topic in this demo video. Okay, so let us start the video. So here, the topic that I have chosen today, that topic is reproduction. It's reproduction. Okay, so when we talk about reproduction, basically, so what is reproduction? It should come in the mind. Okay, so that means the word itself is having that two terms basically, and those are re and produce. Means the organisms are producing their individuals, okay, offsprings, okay, which are similar to them, which are similar to parents basically. Got it? So here, if we talk about reproduction, so reproduction is what? Producing individuals of its own kind. Okay, and you know that this reproduction is very important. Now, Mike, if I raise this question to you, that is it important for individual or is it important for a species? So the one thing that comes very commonly in mind, array, why not? Why not? It's important for individual also. But when we talk about the survival, okay, of that individual and the species. So reproduction doesn't play any important role in the survival of the individual. If the individual organism is not going to reproduce for the whole life will be normal. But it is very, very important and vital process for species survival. Because a species is what? When we talk about a species generally, so what is what? It's a group of individuals which can interbreed among themselves and can produce fertile offsprings. This definition was given by Ernst Mayr. Okay, so if we talk about here, so it is very, very important for a species survival. And it will help in the maintenance of the population of that particular species on this earth. Okay, fine. Now coming to the third point, means when we talk about reproduction, so we have studied reproduction's two modes. One is asexual and one is sexual. So you will be surprised at what we are going to take in this video. So for this video, I have chosen a class 12 chapter, not the whole chapter basically, but a topic from it, okay, and that is sexual reproduction in flowering plants, the chapter I have chosen for this video. So let us start with the sexual reproduction in flowering plants. Okay, sexual reproduction in flowering plants. Because we have the basic idea. Okay, when we talk about the sexual reproduction in which the two parents are involved, they produce the gametes, and then gametes fertilized, it produces the embryo, and then embryo produces the individual. Okay, the whole events are there basically. So when we are stick to class 12, it means here you can say what we generally teach basically in class 12. So the idea of sexual reproduction is there in the mind. So in this video, we are not going to discuss those events, but we are going to take one or two topics from it. Okay, so when we talk about this chapter is also important from NEET point of view. When we talk about this chapter from NEET point of view, okay, so if you calculate from 2017 to 23 basically, means you can say 5 to 6 years, okay, 6 years. So from this chapter, NT has asked around 20 to 21 questions. Means it's hold about 2.6%. The percentage weightage of this chapter in NTS is according to the questions which have been asked in previous years. Okay, so from NEET point of view, means you can say that 2 to 3 questions every year comes, okay, from this chapter. Every year come from this chapter on an average, if we talk about. So this is very important chapter basically. Got it? Now, when we study this chapter, so we study certain important, you can say, topics. First, we talk about the structure of flower so that it will give you an idea. Okay. Then we talk about what? Pre-fertilization. Okay. Where we talk about means structure and events. Where we talk about pollen grains, where we talk about, okay, you can say embryo sac, means pollen grain formation, structure, embryo sac structure, formation, when we talk about this with certain other topics also, subtopics. Okay, next, when we talk about the next topic here, so we talk about double fertilization. Double fertilization is the main topic. But obviously, when we talk about double fertilization, prior to that, we talk about the pollination process, then we talk about pollen-pistil interaction, all these things. Okay. After that, we take the post-fertilization events. Okay, post-fertilization events, where we talk about endosperm, and then we talk about seeds, embryo, then we talk about seeds. Okay, and finally, we take two topics, and that is what apomixis and polyembryony. These are very interesting topics basically. But the question is that, are we going to take the whole topic today? No, not at all. Okay, so I'll take two topics basically. One will be the structure of the flower, and one will be from the pre-fertilization events. That will be, you can say, we will talk about the anther, the transverse section of the anther, the detailed structure of it, and then pollen grain we will talk about. Okay, so let's start with the structure of the flower. Structure of flower. So now here, two things are there. When a common man sees a flower, okay, so he sees a flower with different values, means flower holds basically the aesthetic value. Okay, ornamental value, we place the flowers, you can say, in our home also. Okay, ornamental values are there, religious values are there. Okay, social value, on many events we take up the flowers and produce as a gift and all, everything. Social values are there, cultural values are there. Okay, but when a biologist sees a flower, okay, when a biologist sees a flower, so what he sees basically, okay, so he sees flower as a modified shoot. He sees as a modified shoot. Now you will say that means, what is the meaning of this modified shoot basically? Okay, so let us understand in a very crisp way. Here, when the flower is formed, okay, means when flower formation takes place, it is not a one-step process. Flower formation is not a one-step process. Okay, flower formation means changes occurs. Changes occurs. And changes, as per the NCERT, it is written in NCERT also, that is the hormonal, or you can say structural changes. Because that part of the plant which is going to form basically the flower, so the hormonal changes will be going on. Those hormones will drive the change which will form those cells, the part of the shoot, into a flower. Okay, so generally what happens when we talk about the changes which will be going on, so the apical part, you can say the shoot, apical part. Okay, so it changes to what? The flower primordia. Okay, flower primordia means what are primordia? Small birds which will form the future flower basically. Now after this, from flower primordia, what will be formed? The floral buds. And then it will form what? A complete mature flower. So we can say that now means the shoot which was present, it has been modified into a mature flower basically. It has been done under hormonal action basically, and which led to the different structural changes which went, you can say, series wise. Okay, now here if we see the structure of the flower basically, so we find different parts, and the flower is generally made up of 4 whorls. We will see it with the help of the diagram. Okay, so here I'm drawing the first whorl for you. Okay, which is known as what? This structure is known as what? The pistil, and it is having 3 parts. This part is known as what? The stigma. The narrow part, the slender part, it is known as what? The style. And the third swollen part, it is known as what? The ovary. Now this is known as what? The pistil. Okay, which makes up, okay, which makes up the gynoecium. Okay, so when we talk about the 4 whorls, so the first whorl which we are talking about here, that is known as gynoecium, which is the collection or which is made by the pistil or the carpels basically, which is also called. And technically this pistil is known as what? The megasporophyll. Megasporophyll. Okay, now when we see this ovary, so what we are going to find here, okay, so here we are going to find the structure, very important structures, which can be one or many. Okay, these are known as what? The ovule, which is going to form our seed basically. Now these ovules are attached to the tissue which is present in the ovary, and this is known as what? The placenta. So in your class 11th also, you have studied about different kinds of placentation. Means placenta is the place of the attachment of the ovules basically. So you have studied about marginal placentation, then axial placentation, then parietal, basal, free central, all these placentations you have studied. Okay, so this is the one whorl of the flower. Amongst 4, we have the one whorl. Now coming to the next one, the next whorl that we will talk about, and that is considered to be the male part. Here we are taking, you can say, a bisexual flower. Okay, means the flower which has both male and female reproductive part. Okay, so here this is our second whorl. Okay, and this structure is known as what? Stamen. This is known as, the whole structure is known as stamen, which is also known as microsporophyll. Okay, microsporophyll. So you will say, so which whorl it is forming basically? So it is forming a whorl which is known as androecium, which is forming what? The male part, and this gynoecium it is forming what? The female part. So the gyne term is associated with the female, and andry term is associated with what? The male. So it is the second important wall which is present in the flower. Now, what is the stamen is made of? It is made up of two parts. One is considered to be the anther, and one is considered to be the filament. One is considered to be the filament, basically. Now, when we see these two whorls of a flower, so what happens, if you will see these are the two very important whorls of the flower. Why? Because they are considered to be involved in the reproduction process, means in production of the gametes, in the fertilization process. So that's why these two are considered to be what? The essential whorls of the flower. They are considered to be the essential whorls of the flower. Essential whorls of the flower. Okay, which one? One is gynoecium and one is androecium. Essential whorl. Why? Because they are involved in the reproduction process directly. So the out of four whorls, two whorls are essential. One is gynoecium and one is androecium. Okay. Now coming to the next two whorls. So here, when we talk about the next two whorls, so we have the next whorl, which is made by the petal. These are what? The petals. And these we know are the colorful parts of the plants. Petals are white also, but generally they are colored. Why? Because they attract insects for the pollination. That we will study when we will, because this is the demo video basically. So when we will study in later, so when we talk about pollination all, so there we understand the role of the petals. So petals are generally what? Forming the third whorl, and it is known as what? The corolla. It is known as what? The corolla. Okay, and coming to the fourth whorl, okay, which are generally green part of the flower, and this part is known as what? The sepal. These are the sepals which help in the protection of the flower, or you can say the reproductive parts, male and female, during the budding stage. Okay, they enclose the flower, they protect it basically. Okay, and this is making what? The fourth whorl, that is known as what? The calyx. Okay, so here we have understood four whorls of the flower, which is gynoecium, androecium, corolla, and the calyx. And here, when we see corolla and calyx, so we generally don't find the direct role of these parts of the flower or these whorls of the flower in reproduction. They are not directly involved, and that's why they are considered to be what? The non-essential whorls of the flower. So in non-essential, we have two. One is calyx and one is corolla. Now here, all these whorls are attached to the flower on a swollen part, okay, which is known as what? The thalamus or receptacle, which is attached by the stalk, through which it will attach itself to this stem. So here what we have studied? We have studied the typical structure of a flower, which is made up of four whorls. One is calyx, corolla, androecium, and the gynoecium. Okay, now coming to the next one. From here, what we are going to pick up basically, we will start with the androecium, and in that androecium we will start with the structure which is known as microsporophyll or stamen. Microsporophyll or stamen. Okay. Now here, if we talk about stamen, it has typically two parts. One is known as anther and one is known as the filament. Okay, so let us see these structures. Let us study these structures in more detail. Okay, so here what I have drawn, that is the anther part, and the long slender structure which is known as what, the filament. So this is the anther and this structure is known as what, the filament. So it is the swollen part and it is the long slender stalk. Now here the question has come in the NEET that the part of the stamen which is attached with the thalamus or the petals of the flower. So this end is considered to be the proximal end of the stamen, and through this end it is attached to the, you can say, thalamus, or you can say the petals of the flower. This question has been asked in the NEET in the previous year. In the one of the previous year, we will see, inshallah. Okay, so here we have to understand this. Now let us take this structure in more detail. Now when we see this anther, we are going to take this anther. So a typical anther, okay, so it has what, two lobes. Typical anther is having what, the two lobes, means it is a bilobed structure. Bilobed structure, and it is having two thicker, okay, which is known as what, diathecous condition. It is known as what, the diathecous condition. Now you will be worrying, what are these basically? Two lobes, two thicker. Okay, so we are talking about what, the typical anther. Few examples we will see later also. Okay, so for this, to understand this, we have to cut the anther basically, and we have to see the anther in more detail. So let us draw it. So when you will cut the anther in transverse section, okay, when you cut the anther in the transverse section basically, so what we are going to get, basically we have to understand this. Now what you are going to find here, see here we have two lobes. Okay, we have two lobes. So this is what considered to be the bilobe condition. This is considered to be the bilobe condition. Now in each lobe, what you are going to find? You are going to find a cavity. See, a cavity which is making further two lobes. Okay, so this is considered to be the diathecous condition, means in each lobe we have further, you can say a cavity which is dividing it actually into further two lobes. Okay, so this is considered to be the diathecous condition. Okay, so two lobes, bilobe condition. In each lobe we have two thicker. In each lobe we have two thicker. See, two thicker is there. Okay, so this condition is known as what, the diathecous condition. So what happens when there is a diathecous condition? So actually what we are getting four actually chambers. You can see here we are getting what, four chambers. We are getting four chambers. Okay, so here we can see that means we can say that this anther which is bilobed and diathecous condition, okay, it is having four chambers which are known as microsporangium. Microsporangium. Okay, microsporangium. So it is having what? The 4 sporangia. This condition we can call it what? Tetrasporangiate. Tetrasporangiate. Okay, so these 4 sporangium, or it is known as what? The microsporangium, or we can call it what? A pollen sac, which is the microsporangium of the anther or the microsporophyll. Okay, so here what we have studied, that in one typical anther there will be 2 lobes. There will be 2 lobes, and each lobe will be having 2 theca. That's why it is what considered to be the dithychus condition. Okay, which is making 4 chambers, which are considered to be the microsporangium, and that's why we have 4 sporangia. Okay, so here we can call it what? The tetrasporangiate condition. Okay, and which in each microsporangium is called as what? The pollen sac, basically. Okay, now the question arises that, yes, one more thing that we have to see here, that here when we talk about the typical anther, it is having, see, it is four-sided structure, that is tetragonal structure. It is having 4-sided structure. Okay, and here what we will find, we'll find the line of dehiscence. Okay, means through which the pollen grains, means it burst from this line, and then pollen grains are released. Pollen grains are released, basically. Okay, fine. Now the point is this: every anther has the same structure? No. Typically, generally, anthers are bilobed and dithychus condition, means each lobe will be having 2 theca. But apart from this, we have monothecous bisporangiate condition also. Bisporangiate condition, like in Malvaceae family. Okay, monothecous means it will be 1 lobe only. There will be no 2 you can say divisions or cavity will be there. It will be forming further 2, you can say, chambers. No, it will be monothecous, 1 lobe. Okay, 1 theca will be there. But bisporangiate means it is tetrasporangiate, so it will be having what? Finally 2 lobes. Here we have 4 lobes. Okay, 4 pollen sac is there. Okay, similarly we have monothecous and monosporangiate condition also. Monothecous and monosporangiate, like in Arceuthobium. It is a parasitic plant, basically. So here we have 1 theca, okay, and one sporangium only, monosporangiate, that is what having 1 theca. Okay, so here if you see a typical anther, we have what? We have a bilobe condition. Again, I'm telling because generally the student get confused. Okay, and in each lobe we have 2 theca. So ultimately 2 lobes are forming what? The 4 microsporangium, which is considered to be the pollen sac of the anther. Okay, now getting into more further detail. Now if we see this in a transverse section, okay, so what we are going to find generally means, if you are going to study these chambers in more detail, okay, so what we are going to find. So if we see that, so here Okay, so we are going to find in every pollen sac a collection of tissues okay, these are known as what? The sporogenous tissues. Sporogenous tissue, and which are diploid. And what is very, you can say, important and beautiful about these sporogenous tissues, that these sporogenous tissues cells, means each cell is a potent cell to form pollen grain. Means each cell of it, or we can write here, each sporogenous cell is potent to form pollen mother cell, which is known as what? PMC. It is mentioned in NCERT. Okay, it is also known as microspore mother cell. It is known as what? The microspore mother cell, which is MMC. But in NCERT, MMC is mentioned for megaspore mother cell, PMC is mentioned for pollen mother cell. You have to take care for this. Okay, so now each cell of it is a micro pollen mother cell, which is going to form what? The pollen grain. Pollen mother cell, which is going to form the pollen grain. Simple. We'll see it later. Now here, if you see this, okay, sporogenous tissue. So this is sporogenous tissue. It is surrounded by a very important layer, okay, which is known as tapetum. Which is known as tapetum. We will study it in further detail. Okay, a very important layer that is known as what? The tapetum. Okay, further, if you see this, now here, this tapetal layer, okay, that is surrounded by, you can say, further layers okay, and these are known as what? The middle layers. These are known as what? The middle layers. Okay, now after this middle layer, let me see here. Actually, I don't have a space here. Okay, so we have another layer. Okay, this is known as what? The endothecium. And finally, we have what? The epidermis. Finally, we have what? The epidermis. And these all, you can say, tissues, they are connected by the ground tissues, which are known as what? The connective or connective tissue. Okay, so when we see our anther in a transverse section, we see this type of structure basically. Now here, this is not sufficient to understand. So let us go through it in more, you can say, further details. Now here, as I have shown you that yes, this is the outer layer. Okay, this is known as what? The epidermis. Okay, now this epidermis will be followed by, okay, the endothecium. Okay, endothecium. Now here, if we talk about epidermis, what it is serving? It is serving the protection function. It is giving a protection function. If we talk about the endothecium, so it is helping in release of pollen. from these anther lobes. Okay, release of pollen grains. How it happens? Because it has the nature, because of its hygroscopic nature. Because of its hygroscopic nature. So it absorbs the moisture and make it what? They dry. Okay, now after this, we have what? Okay, we have what? The middle layers, which is generally 1, 2, 3, and it degenerates later. Okay, and then we have a very important cell layer, okay, which is having okay, which is having what? The cells which are, you can say, binucleate as said by NCERT. ऐसा क्यों होता है? उनके पास multiple nucleus क्यों होता है? Because karyokinesis is not followed by cytokinesis. दूसरा, they have dense cytoplasm. They have the dense cytoplasm. And this is very, very important. This layer is very important. Why? Because it nourishes okay, the microspore mother cell or pollen mother cell. Okay, it nourishes it. Now here inside, means inside to this layer, we have what? The cells which are known as what? The microspore mother cell. Okay, microspore mother cell. So each sporogenous cell, just the same thing that I told you, that each sporogenous cell is a potent microspore mother cell. Now this microspore mother cell will undergo changes, which are undergo divisions to form the pollen grain. Understood? So microspore mother cell, it is also what? The 2 in here. It is a diploid structure. It is a diploid cell basically. Now this tapetum will provide nourishment to this microspore mother cell so that it can go under divisions and can form the pollen grain. Okay, so here also means this the question has come in the NEET also, means in the previous years, that tapetum, what it does, it provides what? The nourishment basically. Okay, so one question has come from here also. Now here, if you are going to see the tapetum, so what tapetum does? Only nourishment? No. It helps in the storage of food. It helps in the transfer of food. Okay, it helps in the formation of sporopollen, and that is a very important component of the outer layer of the pollen grain. The formation of sporopollenin. Okay, and it helps in what? The formation of pollen kit material. Pollen kit material. Pollen kit material, they are this, you can say, that is one component is the lipid and one is the carotenoid. Now pollen kit material, that are means that surrounds the pollen grain in generally those flowers which are assisted by, you can say, the insects for the pollination, so that they can stick to the body of the insects basically. So this tapetum holds a very important role. Okay, understood? So here means when we talk about the tapetum, so we have different, you can say, different types of tapetum which were found in, you can say, previous or you can say primitive angiosperms, and advanced type, you can say, angiosperms are there. So that was amoeboid, you can say, tapetum, or invasive type, or glandular tapetum, where it, you can say, how it takes up, you can say, the nutrients from the middle layer and then transfer it to what? The microspore mother cell. Okay, pollen mother cell. Okay, so here, moving further, so what is going to happen? That these microspore mother cell, okay, these microspore mother cell, or pollen mother cell, which is said by NCERT as PMC, they undergo division and they form what? The microspores. Microspores. This will be 2n and this will be n. So 100% there will be what? A meiotic division will take place. And this is known as what? The microsporogenesis. And then each microspore means, because these microspore will form what? They will be in the form of a tetrad, microspore tetrad. Okay, microspore tetrad will be there. Okay, so from the tetrad, each microspore, that will form what? The pollen grain. That will form what? The pollen grain. Okay, and with the help of mitotic division, that can be 1 and then 2. Okay, we are not going to take now more further detail, otherwise the video will be longer. So mitotic divisions will be there to form what? The pollen grain. Okay, so this step, the formation of pollen grain from microspore, that is known as microgametogenesis. And this pollen grain is known as what? The male gametophyte. So microsporophyll, that is stamen, microsporangium, that is pollen sac, and male gametophyte, that is the pollen grain. Okay, so here I'm stopping the video. Okay, hopefully I hope that through this video you can say the content has been enjoyed during the whole lecture. Many things that I have taken here, okay, that is according to the topic that I have taken, or chapter what I have chosen for today's demo basically. And hopefully we cannot conduct, or I cannot teach the whole chapter in one go, okay, for this video, otherwise the video will be more longer. Okay, so hopefully you might have enjoyed this video. Okay, thank you very much.